I'm really excited to talk about this paper with you guys because I'm an author on the paper, which means I'm officially a psychedelic drug researcher along with my PI, Robert LaPrairie. The collaborators for this paper were Ellen and Christoph at the University of Ghent in Belgium, and both Vadim and Olga, our Russian collaborators, synthesized the compounds that we used in this study. Let's go over a little bit of background information. So NBOMe compounds are a potent class of psychedelics that act at serotonin 2A receptor. This is the structure of them. A lot of you have probably heard of NBOMe compounds because of their high extreme potency. Psychedelic compounds are now being studied for their therapeutic potential to treat psychological disorders such as post-traumatic stress disorder and depression. The neuropharmaceutical company MindMed or Mind Medicine is currently running a 1-year phase 1 clinical trial testing a combination of LSD and MDMA for the treatment of major depressive disorder. With this being the case, there is a demand for the creation of novel psychedelics. In this study, we sought to understand how the position of the methoxy groups affects 1. The NBOMe compound's activity at serotonin 2A receptor, and 2. The binding site of the NBOMe compound at serotonin 2A receptor. Here's the general structure of an NBOMe compound. It has 2 parts. It has this phenyl ring, which is labeled 1 through 6, where we could put different methoxy substituent groups. And the right-hand side of the molecule is called the N-methoxy benzyl part. The NBOMe compounds we studied in this paper were synthesized in Russia by our collaborators, Vadim and Olga. So the first compound they made for us is this 25H-NBOMe compound, where you have the methoxy at the 2 and the 3 position. Then we have 24H-NBOMe, where we have methoxys at the 2,4 position. 2,5, where we have methoxys on the 2 and the 5 substituent on the phenyl ring. 26H-NBOMe, 34H-NBOMe, and finally 35H-NBOMe. So we're really trying to look at what differences do placing these substituents at different positions on the phenyl ring mean in terms of the structure-activity relationship of the NBOMe compound. The in vitro studies for this publication were done by Ellen at the University of Ghent in Belgium. In this first panel, let's take a look at the psychedelic NBOMe compounds at serotonin 2A receptor in terms of their activity. What we can see in panel A is a graph, and on the x-axis we have the drug concentration or dose, and the y-axis we basically have the response that that drug exhibits. The further shifted to the left and up, the higher the potency and the efficacy of the drug, and the further shift down and right would be a lower potency and efficacy. We can see that LSD in purple is used as the control compound, and we can see in B that it has an EC50 of 4.44 nanomolar. And then we can look at all of the other compounds. The biggest noticeable thing is that the drugs with the highest potency are 25H-NBOMe as the curve goes all the way up, and also the psychedelic compound 24H-NBOMe is also very potent. The general trend here to notice by panel B is that when we have a 2 substituent, so 25, 23, 24, 26, the potency is relatively the same. But when we change that to a 3 position, as in 34 or 35H-NBOMe, the potency drastically decreases. So we're going from about a potency of, let's say, in the case of 26H-NBOMe to 23.9 nanomolar, and 34 and 35 we're going to a potency of 513 and 485 nanomolar respectively, indicating that there's a drastic loss in potency when you change the 2 substituent to a 3 substituent methoxy, and I find that quite interesting. Because we saw this drastic shift in potency when we changed the methoxy group from a 2 to a 3 position, decreased so much, we wanted to look at in silico as to why this might be the case. Can we figure this out from how it docks to the receptor? Is there something special about the 2-position methoxy that doesn't happen or occur with a 3-position methoxy? So I did the in-silico studies for this publication, and I could not have done this without the crystal structure of serotonin 2A bound to 25C and NBOH, which was produced by the Roth lab, or Brian Roth. So thank you, Brian Roth, for the 6WGT crystal structure that allowed me to do this modeling and make it nice, fluid, and easy. Here is the serotonin 2A receptor. It sits on a plasma membrane, separating the inside from the outside of the cell. Part of the receptor sits in the extracellular space outside the cell. Part of it sits in the intracellular space inside the cell. It has an N-terminus, and it has 7 transmembrane helices that cross that plasma membrane. 1, 2, 3, 4, 5. If we spin the receptor around, 6, 7. Helix 8 isn't really considered a helix that crosses the plasma membrane, but I like labeling it just for complete picture. The first step in this in silico process is building these NBOMe compounds. This first one is 25H-NBOMe, and if we spin it, we can see that the methoxy groups are on the 2 and the 3 position of the phenethylamine ring. We can see them right there again. The next NBOMe compound I built was the 24H-NBOMe, where we can see the methoxy groups are on the 2 and 4 position of the phenethylamine ring. This is quite easy to do actually if you have the right chemistry software. The software I use to build these is called Spartan by Wave Function. And 25H-NBOMe, where we have the methoxy groups in the 2 and the 5 position. I will not include the other NBOMe compounds because that is quite redundant, but I hope you get the idea of what's going on there. Here is just a visual showing how the NBOMe compounds dock to serotonin 2A receptor with the multicolored helices and the compound bound to the orthosteric binding site of the receptor, and the drug is in this light blue-ish color. Let's look at how the NBOMe compounds bind to serotonin 2A receptor. This is the view of the receptor from the N-benzyl moiety and the interacting residues around that N-benzyl moiety and how it's stabilized inside that part or binding pocket of the receptor. This is an interacting residue chart, and basically the more negative the number, it means that that interaction is stronger. The most important interaction in psychedelics binding to serotonin 2A receptor is the interaction between aspartic acid 155. Specifically, it's a salt bridge. It's a really strong interacting residue, and as we can see, the interaction between LSD and all of the NBOMe compounds are relatively the same because they all form that salt bridge, and they usually form a hydrogen bond also. There's also a nice strong interaction with serine 159. As we'll notice, the interaction with LSD is quite weak, and the reason for that is because with the NBOMe compounds, there's a hydrogen bond, making the residue interaction energy negative 9.34 kcals per mole, but with LSD, this hydrogen bond does not exist because you can't have a hydrogen bond at that position in LSD. Okay. There's also a strong interaction with tryptophan 336. The interaction with LSD is only negative 2.3 kilocalories per mole, whereas all the NBOMe compounds, it's about 7 to 9 kilocalories per mole. The next interacting residue is phenylalanine 339, which is strong amongst all psychedelics. And the last one is phenylalanine 340, which once again is a pretty strong interaction amongst all psychedelics. These phenylalanine 340 and 339 are actually the two residues that stabilize serotonin 2A receptor into its active state where it is activated and can produce psychedelic effects. In this photo, we're looking at the psychedelic NBOMe compound 24H-NBOMe from the viewpoint of the phenethylamine, not the N-benzyl. And we can see this is where we put the methoxy substitution groups. So if we label the ring, 1, 2, 3, 4, 5, 6, this is 24H-NBOMe. The position on number 5 should be white, not red. VMD was giving me some problems with this. I don't know why, but that is okay. We'll roll with it. We get that interaction residue table as we talked about before, and we'll look at the strength that the methoxy group interacts with a specific amino acid residue. So 2 position interacts with serine 159 and threonine 160, and we can see this by the interaction residue energies on the table. Anything with a 2 position interacts stronger than anything with a 3 position at threonine 160. Same thing is true at serine 159. When we change from a 2 to a 3, it drops to about 8 kilocalories per mole from 9. Then we have glycine 238, which interacts with a 3 position methoxy. It's actually glycine 238 and serine 242. For glycine 238, we can see with a 3 position, there's a stronger interaction there than if we change that to a 2 position, it drops off, just because it's further away in the binding pocket. For serine 242, same situation. 3 position has a stronger interaction than anything with a 2 position methoxy. 4 position interacts with serine 239 and valine 235. They're located here. There is serine 239 and there is valine 235. So anything with a 4 position is highlighted, and when we change that from anything other than a 4 position, it drops off. Same is true for valine 235, and then it drops off. And then asparagine 343 I have in this table too. That also interacts with the residues, but it's not as apparent. It's only apparent that the 5 position methoxy interacts with this residue. And then anything without a 5 position, it drops off. And the conclusion of this paper is as follows. From the in vitro studies, we can see that NBOMe compounds with a 2 position methoxy display greater potency and efficacy than a 3 position methoxy. From the in vitro studies, we can also see that compounds where the methoxy groups were next to each other, 23 and 34H-NBOMe, the potency and efficacy is reduced. This is kind of interesting because if you think about it from a chemistry perspective, if you put 2 bulky substituents such as methoxy groups next to each other on a ring, in order to avoid steric clashing, they have to orient themselves 180 degrees away from each other, okay? As opposed to if they are placed with 1 carbon in between, this is not as much of an issue. In-Silico Studies showed that 2 position methoxy interacts stronger with serine 159 and threonine 160, both on helix 3. In-Silico Studies showed that a 3 position methoxy interacts strong with glycine 238 and serine 242 on helix 5. A mounting research shows that movement of helix 5 is important in activation, so we think this difference in the interaction with helix 5 leads to the difference in activity of NBOMe compounds, although you'd really have to set up a pretty wild molecular dynamics study to prove this, but that is just a hypothesis. I hope you enjoyed this publication review.